All right, so ladies and gentlemen, we are going to discuss designing experiments and parts of experiments. So our expectation for students in biology is that you can tell us the steps of the scientific method, and you can identify the different types of variables, independent and dependent variables in an experiment, as well as controls and constants. So we're going to start with the scientific method. This is the same thing you've been taught your whole time you've been in a science class. So scientific method, that's what the SM stands for, is a process used by scientists to find answers to a question. The general order of steps: make an observation, state the problem, make a hypothesis. Remember, a hypothesis is a fancy word for educated guess. Design an experiment, conduct the experiment to gather data, analyze the data. Usually that's going to be in the form of a graph, and then write a conclusion. All right, so let's move on to the variables in an experiment. A variable is anything that could affect the outcome of an experiment. The two types of variables that give kids the most trouble are independent and dependent variables. Independent variables is the variable that the scientist changes. So when you go to identify the independent variable, you want to look for the intentional, which means I meant to do that, difference. Okay, so the independent variable is the difference that the scientist chooses. Okay, the dependent variable is the variable that responds to the independent variable. When you go to identify the dependent variable, you're going to look at the data table. So the dependent variable you can identify because it is the observation that got recorded in the data table. Okay, and then constants are variables that do not change throughout the experiment. Okay. Experimental versus control group. The experimental group is the group that gets the independent variable. So this is the group that gets the difference. The control group does not get the independent variable. So the purpose of the control group is that it is used to compare how the test subjects responded to the independent variable. Sometimes there's a control, sometimes there's not. Sometimes you're just comparing a whole bunch of different, well, not a whole bunch. You're just comparing different amounts of something where there is no control. So the control can be present or it cannot be. So it's not necessarily something that is always present. So here's an everyday experiment before we go into a biology one. So it says, Tony watches his grandmother make bread. He asks her how the bread dough rises. She explains to him that as the yeast consumes the sugar, it releases carbon dioxide, CO2, gas, which causes the bread to rise. Tony thinks that if he uses more sugar, the dough would rise higher. To conduct his experiment, he makes 4 different bread doughs according to the chart below. All the ingredients are the same except the amount of sugar. Since his grandmother always used 50 grams of sugar, he named dough B as the control group. Okay, so if we look here, we've got 4 different types of dough. Dough A got 25 grams of sugar, dough B 50, dough C 75, and dough D 100 grams of sugar. Okay, so the amount of sugar is what the scientist, in this case Tony, has decided to manipulate, to make it different. And then he measured how high the bread rose. So based on the information, what is the independent variable? Well, the independent variable is deliberately, let's see if I can spell that, D-E-L-I-B-E-R-A-T. You're going to see throughout the year that I am a horrible speller, so that could be wrong. So the independent variable is deliberately different. Okay, so in this case, it's going to be the amount of sugar. Okay, because Tony put the different amounts of sugar into the different doughs. Now the dependent variable is going to be what he observed. So the dependent variable is his observation. So in this case, what did he observe? Well, he observed that dough A rose 50, dough B rose 10, dough C rose 12, and dough D 15. So the dependent variable is the height of the bread. And then what are some constants that Tony would have to use? So to keep things fair, what would he have to use? He would have to use the same type of sugar, same oven, same temperature setting. He would need to use the same yeast. Yeast can expire, so you wouldn't want to give dough D brand new yeast and dough A some old yeast that you had sitting around. So those would be some constants that he would need. All right, so let's move on to a biology experiment. So a student hypothesizes that plants grow best in green light because they're green after all. The student obtained 4 bean seeds and plants them in identical containers in the same type of soil. The student places 1 plant under red light, 1 plant under blue light, 1 plant under green light, and 1 plant under white light, which is regular light that we have in our classroom and we have outside. So the students measure how tall each plant grew after 4 weeks. Okay, we don't have a data table here, so you just have to look at the description. What is the independent variable? So the independent variable is what was deliberately different. Okay, so what was deliberately different in this scenario was the color of light. All right, the next question, what is the dependent variable? The dependent variable is what the student is going to observe about the plants. So it tells us that the student measured how high or how tall each plant grew. So our dependent variable is going to be the height of the plant. Okay, which plant is the control? Okay, so remember the control is our standard for comparison. So in this case, where do plants normally grow? A plant normally grows outside in regular or white light. So for that reason, the white light would be the control. And then what are 2 constants? There are lots of constants in here. So the containers, they tell us that they are the same. Containers. They're bean plants. They all grew for 4 weeks. I ran out of room, so it's underlined that. Containers, the type of soil is another constant. So you only had to name 2, but you can see that there are 4 that we that were named specifically in the description, we could also assume that they each got the same amount of water. Because if one plant got more water than another, that could throw off the results. Well, it would throw off the results. All right, all right, ladies and gentlemen, I want you to practice this experiment on your own. So it tells you in biology lab, you perform an experiment to test the effect of pH on the rate of enzyme action. You use enzymes found in laundry detergent. You know the enzymes in laundry detergent will eat, quote unquote, gelatin. You pour 25 milliliters of gelatin in 4 test tubes. Once the gelatin solidifies, you place a small marble in each test tube. Next, you fill each test tube according to the table below. So you have 4 test tubes that each look like this test tube that I just put a box around. All right, so they each have 25 milliliters of gelatin, which is, the gelatin is this, well, that was supposed to be blue, but it's red, okay? So 25 milliliters of gelatin at the bottom. And then they are each going to have the same amount of solution at the top. Now the solution is going to be in tube A, just regular water, no enzyme. So here in tube A, no enzyme. Okay, tube B, the solution is going to have a pH of 2. Okay, so pH 2 is the pH of the solution. Tube C, solution has a pH of 5, and tube D, solution has a pH of 12. Okay, so once the 4 test tubes were set up, each with their different solutions, you measure the distance that the marble fell down. Okay, so let's say the marble fell down to here. Was measured after 30 minutes. So after 30 minutes, the distance from the bottom of the test tube to the top of the marble, that distance was measured. So given that information, what I'd like for you to do, just jot down what you believe to be the independent variable. So I want you to ID the independent variable. I would like you to ID the dependent variable. I would like for you to tell me there is a control. I would like you to tell me which test tube is the control test tube. And then I would like for you to name one constant. All right, and we will go over these tomorrow in class and do some more work with identifying independent and dependent variables and answering questions that you may have. Alrighty, see you tomorrow.